Hi everyone, I'm Professor John Rasko.
And I'm Dr. Chuck Bailey.
And we have a problem we need your help with. What we're holding here is a representation of the DNA that you've all heard about in your cells. And there's actually a packing problem in your cells because the DNA actually reaches out 2 meters if it was stretched end to end. But when it's packed into the nucleus or the inner part of a cell, it actually compresses down into just a small part of less than a hair width. It's like spaghetti packed into the nucleus of this central part of the cell. And what we now understand is that it's in a very highly ordered packing system. All this spaghetti is specifically ordered. It's not a mismatch. Now, what we have learnt in our research is that there's a particular molecule that we've been studying for over 10 years called CTCF, which is mutated in many different types of cancer, including breast cancer, ovarian cancer, a whole bunch of others. And CTCF structures the DNA in the nucleus and allows that order to be created. Our research is using highly advanced technologies in order to understand the basic biology of that, which we know goes wrong when cancer occurs. So our research is basic and translational, which we hope ultimately, in understanding these structures, we will be able to interfere with them in cancer cells specifically and allow new therapeutics. So Chuck, why do you think this research is vitally important?
This research is vitally important because we are attacking an Achilles heel in cancer, which is this structure that John showed you. We have identified a way that we can weaken that structure to kill cancer cells. Furthermore, the funding from Tour de Cure will help to support the salaries of scientists in our laboratory, help to buy the consumables that we need for our research, and furthermore strengthens that relationship that our lab has enjoyed for many years, over 6 years, with Tour de Cure in being committed to finding a cure for cancer. This has involved us attending Tour de Cure events to speak, to talk about our research, to raise awareness with donors. I've personally been involved in writing some of the events that Tour de Cure has organised.
I think altogether we are strongly committed to seeing a cure for cancer, in this particular case using some of the tools that we've developed for basic understanding of cell and cancer biology, and in so doing we hope ultimately to provide kind of ways of interfering with DNA structure and organisation that will lead to cures for cancer.